Well, you might have noticed that I'm all dressed in black, and that's, to me, the official MECFS uniform. And the reason for that, is my son is severely ill with MECFS. To go into his room, we have to dress all in black. And the reason we have to dress in black is so he doesn't have to use the mental energy to interpret the color of our clothes. And that's too much for him. So I've gotten used to wearing black. I have a black outfit to go into his room, and I decided to wear it to this meeting because that's the official uniform. Now, because my son is so sick, I'm very dedicated to trying to figure this problem out. And I really appreciate other people like Robert Fair, who's willing to really help this. He's indispensable. But what it's like to try to do this, it's like being dropped into a maze. And you have to try to get out, which would be equivalent to curing the disease. And you keep coming to crosswords. Do I go left or do I go right? You can't do both. And so you try to navigate what you work on and how you proceed. So I'm going to take a talk and try to show that problem and also why I make decisions that I do. And it does not mean that I'm right. So if I say I'm going to do this and not that, it doesn't mean the other thing must be wrong. It has to do with my experience and perceptions that this path has a better chance of working than that one. And that's all it means. And it's not clearly that's right. So if I say I'm not going to do something, it doesn't mean that I think it's wrong. It just means it's probably wrong for me to work on it. So one way to try to think this through is that we're pretty confident that it usually starts with an infection. So we need to think about what happens with the start of MECFS. And we do see there's a large number of viruses that seem to possibly cause this, but there are reports that bacterial or parasite infection like Giardia can cause this disease. Years ago, I worked on a large trauma project with Ron Tompkins. And in that, we were looking mostly at auto accidents. And auto accidents appear to be able to cause MECFS. There's also evidence and reports that childbirth can cause it. I don't know the publications. These are just from doctors. But also, I think there is a fair amount of documentation that vaccination can cause this, as well as surgery. All of these can induce the innate immunity. And that makes me take the road of it's innate immunity is the problem. And that's what we should be working on. I should be working on. So it's the innate immunity maybe that is really the problem. Now, one of the components of the trauma project that we worked on is that if you have an auto accident, your microbiome changes very quickly. Now, the microbiome change did not cause the accident. It's something that is associated with it. So that has made me think that I shouldn't work on the microbiome. Doesn't mean it's wrong. It's just making the guess that it's probably not the right thing to do. Now, there is one experiment of one, which was my son. I've written this all out, so if you don't understand what I'm saying, but I'm going to say the same basic thing. So when he got very sick, he could no longer eat. And of course, that's a crisis when you don't eat. You have to do something pretty quick. And probably the right thing to do is put a J tube in and tube feed him. He refused to do that because he did not want to go to a hospital. But we arranged to have a technician come in to his room and put in a PICC line and make a sterile field. That's a hard thing to do, but that's a way to save his life. So we put a PICC line in, which is feeding him through the vein with sterile nutrients. It's not a perfect solution, but it works. When we did that, and we were working with Metabolon, who does the metabolomics, we were sending samples from my son. They did a fantastic job. I think he was their first patient. They told us they would do any sample for free anytime we wanted it. And I sent samples after we put in the PICC line. I got an emergency call shortly after that from the CEO of the company saying there's something dreadfully wrong with your son. And I said, what? He said, all the metabolites from his microbiome are no longer present. And I said, oh, we put him on a PICC line. Oh, he said, thank God, that explains it. And what that means is basically, that doesn't mean this gut is sterile by any means, but it does mean there's very little metabolic activity. So if the microbiome is causing this problem, you would think that some of it's coming through some of the metabolites that the microbiome is making. Well, then the second control for that is that after a year and a half on the PICC line, we convinced him, his doctor, in fact, Bob Navio did most of this convincing, that he had to put in a J tube. And he finally agreed. In fact, that's when we discovered that I won't go into that. But after putting in the J tube, and we had to worry about getting him the refeeding problem, and Bob Navio helped us with that a lot. But then the microbiome came back. Now, when the microbiome sort of went away, there was no change in his symptoms at all, or activity. When the microbiome came back, there was no change in his symptoms or activity. So that's an N of one, but it convinces me that the microbiome could easily be a part of the problem, but it's probably not the whole problem. So we know that the microbiome can make components that are very important for you. We know the microbiome can detoxify things that are in your blood. We know that it can make things that are neuroprotectants. There's even some evidence that it can make BH4. So I'm not working on the microbiome. Now, is this an autoimmune disease? Well, there are definitely autoantibodies. But again, it's coming back to the auto accident. When you have an auto accident, you make a lot of autoantibodies. And those autoantibodies will go away when the person heals. So, so that can kind of convince me also that, yes, the autoantibodies can be causing some of the symptoms, and yes, they should work on it, but it's not probably causing the disease. If you got rid of all the autoantibodies, it's a good chance that you'll still have the disease. However, I do want to show one example of something we discovered that deals with autoantibodies that may be relevant for treatment. And this came up with a question that it's not uncommon to have a patient diagnosed with ME/CFS, and then later they find out that, oh, they know they have MS, or vice versa. So a patient came to us and said she was diagnosed with MS, but in fact she had ME/CFS. And I'm going to cover that in a moment. But that gave us an idea that maybe we should be looking at a reaction that's probably present in many of the MS patients, and that is a catalytic activity of an antibody. And that catalytic activity is being able to cleave the myelin basic protein, and that's how you can destroy the myelin sheath. So we set up an assay to do that, and this is the example of that. And this is a healthy person. Here's the myelin basic protein. We don't see any degradation, but here are 4 examples from ME/CFS patient, four ME/CFS patient examples, excuse me. And what you can see are breakdown products of the myelin sheath in all cases. So now this is not a FDA-approved test. We can't diagnose this person has MS, but it makes it very suspicious that they do have. Now it's also clear that this person has, these patients have ME/CFS. They were diagnosed by an experienced doctor. When I've asked a few physicians about, could you come down with MS and ME/CFS at the same time? And the answer is, of course not. You can't initiate 2 diseases at the same time, and I think that's probably wrong. So this is an example of one of those autoantibodies that were probably made because of ME/CFS that actually has another disease component. And there are several of these possibilities. And yes, that's important to know because you want to be able to treat the patient for this disease. So our estimate is it's still a pretty small sample, and we're going to continue to do more and also make sure and validate this test and get some physicians that know MS to help us with this. But it's right now just an experimental observation. But it's about 25% of the patients have this. So it would be significant to work on it. Now another drug that came up with a patient, and we get a lot of good information from patients, and we listen. So this patient asked Hector Bonilla, who runs the clinic for ME/CFS, for a prescription for Abilify. And when asked why, she said, well, it really helps me. So he did, but not only that, but he decided to try to treat some of his other patients with Abilify using very low dose. Laura Crosby was involved in this and carried out all those studies and the analysis of the data. And then the results for that are, now this is not a double-blind study. This is just asking the patients the effect of it. In some cases, you actually saw some real evidence, like one of the patients was in a wheelchair when they got the Abilify, and then after treatment for a while, they came walking back into the clinic. So the guess was, from just analysis, around 75% of the patients were benefited by this, but not everybody. It's thought that Abilify probably increases dopamine. And that raises the issue of looking maybe at other drugs that affect the dopamine levels. Now, because of the dopamine effect, we decided to look more deeply into some of the metabolism that's going on. And dopamine is made ultimately from phenylalanine. So phenylalanine gets converted to tyrosine, that's another amino acid, and that reaction is an oxidation reaction, but it also requires BH4. And that's a compound that looks very much like folate, but it's used in what's called reduction oxidation reactions. And it's a strong reducing agent that can carry out chemical reactions. And that chemical reaction is for phenylalanine to tyrosine, and then tyrosine gets converted to dopa again, by a repeat of this reaction, also requiring BH4. And in addition to that, serotonin is made from tryptophan, and the tryptophan is the same reaction, converting it to hydroxytryptophan, which then is converted to serotonin. So both serotonin and dopamine require BH4. And so those seem to be important components in this disease. The suggestions are that serotonin is too high, and that suggestion comes from the fact that if you give them a serotonin uptake inhibitor, they get worse. But there's another reaction which I'm thinking is probably more important for this disease, and that reaction is taking arginine. You notice these are all amino acids. I mean, that's often the starting material for a lot of the things in your body that are needed. So arginine is carrying out the same reaction as these, but then it goes to citrulline, and in the process of that, generates nitric oxide, NO. So nitric oxide is a very simple compound, but it's used as a signaling agent in humans, and it's used to allow dilation of blood vessels. It controls blood flow, but it also controls lots of other things. So this has given us information that we're going to now look at BH4. BH4 is hard to work on. If you do a blood draw, you have to stabilize it within 5 minutes of the blood draw, or it'll get oxidized and you won't be able to analyze it. So it's not an impossible thing to do at all. It's just that it's inconvenient. You don't send it off to a laboratory easily to do the analysis. But there's another thing that explains this, and that's another reason for us looking at this. So, and this is not our work, this is from the literature. But what we have noted in patients, that they're usually low in iron, manganese, and copper. And usually we do this analysis by just doing hair analysis because it's so easy to do, and you simply take the hair and you put it through a mass spectrometer and you get the quantitations of these things. And you're looking for the loss of things, not a high level of things. So people often worry about contamination, but they shouldn't worry about decontamination. As you don't get rid of something because it's been exposed to something. And so I think these are valid in hair analysis, and it's very simple. And in fact, we have sometimes patients send us hair samples and we analyze them. And they just put a little bit of hair, like a gram of hair, in a little plastic bag and mail it to us. So it's a really easy assay to run. So these three metals get into cells by a transporter. The problem is that transporter doesn't work unless it has been nitrosylated with nitric oxide, and that reaction requires BH4. So this would explain why these metals are low, in fact, in almost all the patients. And that suggests that nitric oxide is not being produced, or that the reduced level of BH4 is low. This doesn't prove it, but it really strongly suggests it. And that's the reason we need to do a much deeper dive in looking at BH4, because that could give us a treatment. But I also want to think about how to cure this disease. Now, it's possible that there is an infection. There's a lot of talk about the infection, and of course, also being a geneticist, I'm concerned about the genetic components, but we have done analysis, there's much better analysis done looking for genetic defects for the disease, and I don't think there are any. And so there are some mutations we found that may make symptoms worse, and we're going to explore those. They haven't been looked at before by anybody else. These are mutations that are not in the coding regions of proteins, so that they can tend to be missed. But infection is an impossible thing to consider. So we have done a fair amount of analysis looking for a pathogen. We started out by doing, looking for the DNA viruses, but and our first experiment was simply take blood, and we took it from my son, and we isolated particles, because a bacteria or a virus is a particle, and small particles. And then we lysed the cells that came out of that and destroyed all the DNA, because the DNA is protected in the particles. And then we sent that off to be sequenced. And we did massive amount of sequencing from those particles. And we didn't find anything. Now, we've done that a second and a third time and still do not find anything. And what we're looking for here is maybe a new virus or a new bacteria. But we didn't find anything. So the other way to do this is to, and maybe in a more sensitive way, is to go after the, we can make a more sensitive assay directly for looking at the DNA viruses and look specifically for the virus. So what we've done is taken, we basically have done all the DNA viruses. And we don't just look for one spot on the virus, because that could give us a false signal. For every virus we look for, we do 10 different regions of that virus. And we look for a positive signal, we have to see all 10 regions. And of course, mostly what we've been interested in is some of the herpes viruses, but we're looking for anything at all, because we don't want to miss something. What we have found in doing that are that some of the patients you can find EBV and HHV6 as DNA, as replicated virus. Not very many, a few percent of the patients. Problem is, when we do healthy controls, we find the same percent. So it doesn't seem to be an indicator that that's causing the problem for ME/CFS. Now, we've also worried about parasites. We've done extensive, the same kind of analysis for the parasites. We haven't found any parasites. One thing that we have done, and it's still in the works, but one patient, again, this patient's coming to us and telling us things, that he thought he had a nematode infection, because you could see little tiny nematodes in the microscope. So in fact, we isolated the nematodes in the lab and made DNA and made probes to see if other patients have this little nematode. It's very easy to miss, and a doctor looking in a microscope would probably not notice it, because it's an incredibly small little nematode. And so we'll be searching more patients. We haven't done that yet, but it's ready to go. That nematode could be causing a lot of problems. And also, we haven't done this yet either, but because the RNA viruses are a little harder to do, because you have to convert the RNA to DNA and then you can amplify it. But we'll be looking for RNA viruses as well. Now, this is just in the blood, and you can look elsewhere. You can look in the saliva and so forth. But so far, we haven't really come up with something that would indicate that there's a lot of viral DNA. Now, what isn't ruled out is that the viruses don't come out and replicate and make new virus particles, but they carry out some expression in the cell that they're in. If they make an RNA or a protein, that's a different problem. That we haven't looked at, and I think other people are going to do that. However, we did do the same kind of experiment, and this is these samples came from Jonas Bergquist, and that is to look for a DNA activation in COVID patients. And so I tried to get these from places in the US without success, because I said, oh, we need all the blood samples we have. And Jonas said, no, he'll send us some blood samples from Sweden. So we got these samples from Sweden, which is technically difficult because you got to get a CDC approval to import them. But it was a little bit of a surprise when we carried out this analysis. And what we found is that 80% of these patients had activated EBV. That's a lot. Now, they also had, the next in line was HHV6. But we did all the herpes viruses, and one of them that was, kind of surprised us was that cytomegalovirus down here was 0. We did not find any, and we were kind of expecting to see that one. But we did see a lot of EBV. There was a revisit later. These were in the ICU. And most of the EBV virus had been suppressed. Now, one reason to show you this is the question. When you get ME/CFS or long COVID, is it caused by the COVID virus directly, or is it caused by activating EBV and the EBV causes it? And I don't think that's a very clear answer for what's going on. And this could be true for other viruses. We know lots of viruses can do this. Maybe all of them activate EBV. And so that's all caused by EBV, not other viruses. Maybe bacterial infections do the same thing. Maybe auto accidents do the same thing. I don't know. We're going to look at that. I just want to come to one other thing that is an old problem, but I want to keep pounding on it until we get it figured out. And that is another hypothesis of Robert Fair's. And that is, and the reason for doing this is that what happens when you get an infection is you release tryptophan, and tryptophan floods the body. And his thought was that you want to eliminate the tryptophan. And that's why you do this, because microbes can use the tryptophan for protein synthesis, and it's a hard amino acid to make. So the tryptophan was of interest because, well, I'm going to show you the next slide. That'll make it easier. So tryptophan, when it's released, and it will go into cells, and there's an enzyme called IDO1, and that converts tryptophan initially to this formal kynurenine, which then gets converted to kynurenine. And kynurenine needs to be made in the cell to help regulate the immune system. So in fact, cancer cells carry out this reaction so that the immune system can't kill them. The other reason to think about this was the fact that you can block IDO1, and this was actually an idea for an anti-cancer agent, since cancer cells need to do this. So they've made some very potent blockers of IDO1, and when you block IDO1, it leads to extreme fatigue. So maybe that's what's causing the fatigue in the patients. So, and what can happen is that when tryptophan gets too high, it can inhibit IDO1, and then you can't make kynurenine. And that was discovered in 1967 in a test tube experiment. Put the enzyme in a test tube, put in tryptophan, you can see it converted, it'll convert to kynurenine. That's an oxidation reaction, by the way, with oxygen. But if you put in a lot of tryptophan, you don't get anything. It blocks it. So it's called substrate inhibition. And nothing has been made of that as far as I can tell since then. But it would be a possibility that you flood the cell with tryptophan because you have an infection, and that will shut down this pathway. And that may be really critical. So how to figure that one out, or basically how to solve that problem. The problem is we know it works in a test tube, does it work inside a cell? doing this in human cells is tough because it's hard to manipulate them. But for us, it's really easy to do in baker's yeast. So what we did is we took baker's yeast, pulled out the gene that can make kynurenine, deleting it. This is also very easy to do. Put in the human gene, put the human gene under control by the yeast control system so we can control the level of it. Take out all other genes that metabolize tryptophan. There's quite a few of them, but that's also very easy to do. And now you have, and then set it up so when you make kynurenine then can get converted to what's NAD, and NAD is required for ATP synthesis. So if you can't make NAD, you cannot grow. So when we do that experiment, we can grow the cells in very low tryptophan. They grow normal. Normal growth rate, just like the parent yeast. But if we put in more tryptophan into the growth media, the yeast stops growing. And it will just simply sit there for a very long period of time until, in fact, the media evaporates. It will not grow. So that says it works in vivo. Now, why we would do this experiment? Because we want to figure out a way to block the blocker, if you get what I mean. We have to unblock it, and that way we can tell whether it, in fact, affects humans. But we want to do this in a patient. This will be a little tricky. So we have to use a drug that's approved for human use and is non-toxic. So we screened 1,100 FDA-approved drugs in yeast and found several that allowed the yeast to grow. And now it's important to make sure of what the concentration is to do that and make sure that it's correct, and then try to figure out which of those drugs would be reasonable to give to a patient such that it will not harm them or give them any kind of side effects. That's the only way I know how to rule this hypothesis out. And what we'd be looking for is there any change in the patient or any change in their immune system. Now, the reason to be concerned about this a little bit, this is just a model where tryptophan comes into a cell and it makes kynurenine by IDO1. A lot of people say, well, this couldn't possibly be important because the liver makes kynurenine, and you simply take the kynurenine and put it into the cell. Well, Rob calculated the transport problem, how much kynurenine could actually be imported, and it's an insignificant amount. The transporter does not work very well on kynurenine, so you just can't get it into the cell. There's another problem, and that IDO1 carries out an oxidation reaction, and like hemoglobin, it has iron. Well, one of the problems we have with the patients is that they are low in iron, especially the women. So without the iron, this is not going to work, and you're going to accumulate tryptophan, and then you're going to shut the enzyme down. So it's a real possibility. So I think it has to get eliminated. So now, the last thing, and you already heard this from Rob, and I just want to amplify that. I think this attack-and-ate shunt idea is right. I think that's the major problem. Now, it's also possible that there are two problems. So we can't get too depressed if we try to prevent this from happening and know that we've unblocked the attack-and-ate shunt, and it doesn't help the patient. There might be something else wrong that happened because of this whole thing. So we will then look for the next problem, if there is one. But I think this is the right path, and I think it is innate immunity that's causing the problems. Now, also, my guess is, and this is just a guess, that this attack-and-ate shunt just is not causing ME/CFS. My guess is it's probably causing all autoimmune diseases as well. So if we can solve this problem, we can probably solve an awful lot of the autoimmune problems. In fact, if you unblock the attack-and-ate shunt, your autoimmunity might go away. That's a fantastic possibility. And that's why it has to get worked on. So this is just that TCA cycle, and this is just another illustration of that shunt, where it shunts off, and you lose the ability to make ATP by oxidative phosphorylation. And I just wanted to make sure and give credit for Robert Farah and Chris Armstrong that have been working on this. But I'm optimistic, because we're getting really close to something that really is probably causing this disease. And there's a lot of good work going out there now as well. I just feel that we're poised for a breakthrough. It may require multiple breakthroughs. But I think this disease is curable, not just treatable. I think it can be cured. And a reason I think that is I've talked to a number of patients that have somehow miraculously, spontaneously gotten over it, even though they've been sick for years. And it turns out one of my graduate students, while she was a graduate student, came down with this, and she became bedbound. She was severe. And she thought that it was caused by her crashing all the time. So she was really, I might call her obsessive compulsive. I believe she could do anything that was hard. And she said she was just simply not going to crash, and she didn't for a whole year. She did not crash. And she got over it. She's totally normal. And I challenge her, yeah, are you sure? Don't you just get exhausted easily? She says, no, I do not. I run 10 miles a day. And so that's a good definition of cured. Now, I don't expect all of you, if you get cured, to go run 10 miles a day. But it does represent that this thing does not seem to cause any damage, which I think also amplifies the idea it's the innate immunity being activated, because you would not expect innate immunity being activated, which is to protect you, would also cause damage. It would be engineered not to do that. So if you can unblock this, you'll become totally normal, is my guess. And that's why it's so important to try to cure this. Thank you.
Thank you, Ron. Maybe one question as well, seriously over time. The frontier? I don't know where the cube is.
While we're waiting, I want to make sure I thank the Open Medicine Foundation for providing a lot of the funding here, and also Vinod Khosla, who's given us a lot of funding, and also all the patients and families and everything that donated money have kept us really going, and all the patients that come in and donate blood. It's been a tough several years with the pandemic. The patients did not want to come in, and I understood that, but now they are coming back and coming into the lab. So that helps us to get started again on things.
Can you shout your question? I don't know where the cube is.
When I was researching into the activation of viruses and bacteria, obviously interferon alpha is one of the interferons activated in the innate immune system, but is interferon beta or gamma implicated at all in the attack shunts?
Can you hear me?
Yeah, so the question is, we've been focusing on interferon alpha, but there are other interferons that are also important, particularly in antiviral responses, particularly interferon gamma. So does this shunt affect all interferons? Is it just interferon alpha?
No, it's just mostly it's interferon. So the shunt, I think, is activated by interferon alpha mostly. Rob is an expert in this.
Interferon beta is just as good, and you probably get both in response to the innate immune response. They both bind to the same receptor on the cell membrane. Interferon gamma is a totally different molecule, has a different receptor. So I think you should focus on type 1s, that includes alpha and beta.
So beta can do the same job as alpha in turning on the attacking shunts. Okay, thank you.
Yep.
But interferon alpha controls lots of things. So some of those other things may be causing some of the symptoms and so forth. So that actually needs to be kind of looked at, and you might be able to shut some of those down. I'm not sure it was clear with what Rob was saying, is that we can only look for interferon alpha in the blood. But if it's not made in the blood and made elsewhere, then what we're seeing is the interferon that gets in some space else like the brain eventually gets to the blood. And then when it's in the blood, it gets eliminated by the kidneys. So it's going to be very, very low level. So some people may look and, well, that's too low a level to have any effect. No, because you don't know what's it like where it actually is made. And it is certainly possible it's made in the brain. And that would cause a lot of problems. So that's why we have to run very, very sensitive assays. And right now, there's only one company that we have found that can do that level of sensitivity, and we're pushing them to do another factor of 100 better. Because it would make the data a lot clearer. The amount of interferon that we're seeing is about the same level as found in a lupus patient. And if you treat a lupus patient with an antibody to alpha interferon, they get better. Doesn't cure them, unfortunately. So this low level is not sort of unexpected.
Okay, I think we've got to stop. So I think if you want to catch Ron afterwards, please do so. But just thank him again for a wonderful talk.
Thank you.